So I want to tell you about our work using big data and knowledge to dissect the autism spectrum. And as most of you probably know, autism spectrum disorder, or ASD, refers to a very heterogeneous set of neurodevelopmental disorders that are all characterized by two things: reduced communication and social abilities, and increased repetitive and stereotypical behaviors. And the autism spectrum is quite wide. It ranges from Sheldon, over here, wait, where is he? Here, to Rain Man, over here. And besides the famous individuals on the spectrum, most of you probably know at least one individual on the spectrum, because now, these days, ASD is estimated to affect one in every, let's see if I can actually make these slides work. It's estimated to affect one in every 59 kids in the United States. And look at this amazing increase in prevalence. It's been a 40-fold increase in the past 4 decades. And I can talk for the rest of my 30 minutes about why. The reasons for this increase, but I want to focus on what we're doing to improve our understanding. Yeah?
Is it increase or better detection?
That's a very good question. And the way we measure prevalence is individuals diagnosed with ASD. And do you want me to invest a few minutes in that?
No, it's just because in many cases these increases are just better detection.
Yeah, I mean, it's better detection and other... Yeah.
I mean, practically everyone who's a scientist is called on the spectrum.
Yes.
So we're all, this room is 100%.
I mean, only us computer scientists, not everyone. And for those of you at MIT, you know that the prevalence is much higher there. But you're right. The definitions of ASD, autism spectrum disorders, keep changing every few years with the introduction of a new manual for its diagnosis. And that brings me to an important point, that the diagnosis is purely based on behavioral symptoms. There's no other disorder that's not a neuropsychiatric disorder that's diagnosed only based on symptoms. You never go to your doctor and say, "Hey doc, I have a headache," and the doctor immediately knows what you have. So that's what we're trying to avoid. It's actually pretty awful, because we know that autism, at least we think we know, that ASD is caused, I don't want to, now I'm starting to watch my words. We think that ASD originates during early brain development, and by not diagnosing it accurately enough and early enough, we're letting critical time windows go by without any intervention. And it's a shame, because we know that it's not refrigerator moms or the vaccines that cause ASD, but actually a very strong genetic basis. And here I'm showing you twin concordance rates for ASD, which means what is the probability, if we know that one twin is on the spectrum, what is the probability that the other twin would be on the spectrum? For monozygotic twins, this is actually working, that is about 90%, very high for complex traits. And for dizygotic twins, it's about 25%. And that makes autism one of the most heritable complex traits. What does it mean to be a complex trait? It means that no single gene causes autism, but actually a combination of genes, acting in response to a set of environmental exposures, give rise to a continuous distribution of autistic traits in the general population. And the individuals on the clinical end of this multidimensional distribution are considered clinically autism spectrum disorder. So when we study this trait and its causes, we'd like to focus on the entire distribution in order to study the entire variance that gives rise to autistic traits, so social abilities, communication abilities, tenderness to focus on details, and model this entire distribution, focus on its entirety, and not just on this end of individuals considered ASD. So a lot has been discovered in the past couple of decades, and today we know that autism, we can pretty surely say that autism is characterized by extreme locus heterogeneity. What does it mean? It means that there are many regions in the human genome that have been implicated in ASD in some form or another. And here I'm showing you 24 chromosomes, and you can see that on each chromosome there are some red and some green dots, signifying some association, linkage, or other implication with ASD. And pretty much we can summarize our findings based on several strata. We like to differentiate between common variants and rare variants. Common variants are those that are found, usually we like to say, in at least, their minor allele frequency is found in at least 5% of the population. And as expected, most of the variants that have been implicated in ASD are rare, and the minority are common. Also, every different type of variation that could have been measured was measured and was implicated. We're talking about chromosomal rearrangements, the earliest types of genetic variations that could have been measured, and then copy number variants, and different types of higher resolution variants such as frame shift, in-frame, microsatellites, missense, you name it, it was measured and it was implicated. So lots of complexity, and within this great complexity, as of a couple of days ago, the most well-curated database of autism implicated genes says that 831 genes have been implicated in ASD with high confidence. And that's likely an underestimate. And still, with this assumption of thousands of genes being implicated in typical brain development and its alteration in neurodevelopmental disorders such as autism, it comes as no surprise that there's more complexity that we can grasp, than we can make sense of. And today, mutations that are thought to be implicated in ASD are found in less than a third of the patients. So obviously, we don't make much use of genetic data prior to diagnosis, and we first diagnose based on behavioral symptoms, and only then have the affected children undergo comprehensive genetic evaluation to try and better understand what variants are related to their specific type. So really amazing complexity, and it's not as complex as what Weinberg has shown us about cancer, but you can compare it to a very complex somatic disease such as cancer, and another example of a complex disease, this is a germline complex disease, ASD. And similarly to what we've learned from Bob Weinberg, here we also have convergence. We have similarities between different types of mutations and different types of alterations, and they are all somehow related. I've shown you a bunch of loci that are implicated with ASD. It doesn't mean that each individual has all of these loci or all of these pathways even altered, but different families have different variants that we think might contribute to their disease. And these variants are related to one another at the functional level, another dirty word which I shouldn't use, and their function is related in several levels. One level is synaptic function. Many genes that we think play a role in ASD have a role in the synapse, and those of you that probably everybody knows, but synaptic development is a key part of the typical human development. Here it is. Immune function is also a very integral component of typical brain development, and same molecules that guide the immune function also guide typical development and morphogenesis. And another pretty obvious level of relatedness between genes implicated in a neurodevelopmental disorder is that many of them play a role in transcriptional regulation and chromatin regulation, shifting between gene expression programs between different stages of brain development. So to summarize our complex problem, we have a common male-dominant disorder. I forgot to mention it, but you were probably able to see it in the movies that I've shown you. Most of the movies portrayed boys, and actually 80% of individuals on the spectrum are boys. So men, you're in great risk. We have no effective treatment. We don't understand the causes of most cases, and therefore can't pharmacologically treat it very well. We have behavioral treatment, but that only works if it's for kids that are diagnosed very early and not for all of them. And that comes to the next point where we have often delayed symptoms-based diagnosis, which is really not acceptable at this time and age. And we have clinical and molecular heterogeneity. But within this extreme heterogeneity, we have emerging subtypes and convergent etiologies.
Can you tell us whether these mutant alleles are of recent origin, de novo, or have they been in the human germline for hundreds of generations?
So that's a very good question, because a lot of recent research focused on de novo mutations. And part of the reason that was a focus is because they were able to focus on de novo mutations, having sequenced families. And at first, it seemed as though there are many de novo mutations implicated in ASD. But the more families that were sequenced and the more other traits that were studied, and de novo mutations were studied in these traits, it seems that there is no increased rate of de novo mutations in proband as compared to unaffected siblings. And latest statistics suggest that it does have a contribution, but not as significant as we thought previously. Also, if you recall, the twin concordance rates that I've shown you suggest that there's a strong heritable basis. So a priori, there's no reason to believe that de novo mutations play a big role, and they can play a role, but not a dramatic one.
Yeah. On your diagram,
Yeah.
you had the red spots on the X chromosome. How does
That's
that
a very good question. Yeah, the X chromosome has many synaptic genes in it. And many individuals thought that because of the male dominance, the solution to our problem would lie on the X chromosome. And actually, lots of intellectual disability genes are X-linked. The X chromosome has been sequenced in and out, and still it explains, it cannot explain many cases. But it's a very good question. Lots of related genes are X-linked. Yes.
So the fact that it's 1 in 38, this is based on some criteria for diagnostic.
Right, yeah.
How dependent is this? It's not like shape of blood cells. It's more
Exactly, it's behavioral. And so if you ask the psychiatrists, they would say that the criteria keep changing, but it's pretty stable. If you look retrospectively, 30 years ago, people that were diagnosed with autistic disorder are today at the extreme end of what we call the autism spectrum disorder. Yeah.
But it can be less or across?
So it's more, now the diagnosis is more sensitive, and that is consistent with the increased prevalence.
I don't mean to slow you down completely,
Please slow me down.
I really believe that we've just pathologized being a male. So that being a male is then no longer considered a normal state.
You think?
No, and I don't like it because I actually tend to like males. If you take the people who would have been diagnosed as ASD by previous criteria, do you come out with a more homogeneous genetic picture than by diagnosing almost all the males in the world?
So you're right that there's a big theory that says that autism is extreme male brain, because it's the typical male behavior, just gone wild, let's call it. And many individuals have tried to focus on those scoring worse on behavioral instruments, because that's the only way that they're diagnosed. I mean, how would you measure doing worse? You have these behavioral questionnaires, and there were no significant differences in the underlying genetic makeup. You can stratify based on IQ, because many individuals on the spectrum are also intellectually disabled, and there are differences there. But then you get into the question of whether you're diagnosing intellectual disability and not ASD. More questions? All right. Okay, so we have this complex disorder, complex problem, and we have emerging subtypes. So our knee-jerk reaction to this type of problem is precision medicine, our solution du jour. And the idea is that just like your smartphone maps, where we try to integrate different layers of information in order to get a true depiction of the real world around us, we will integrate different layers of molecular, cellular information to understand the altered molecular and cellular mechanisms. So here I'm showing you an example of the real world, and we would integrate cities, streets, parcels, land use, elevation, etc., different sources of information in order to actually get a true representation of the real world around us. In medicine, we've been practicing that for centuries, right? There's never one specialist that you go, there's never one physician that you go see. There's always a specialist, and if you have a complex problem, you use several specialists, and they hopefully coordinate among them, but usually they don't. And there are specialists of different tissue systems and organs, and that's not new. The new thing is that now we're moving from the tissue level to the cellular level, and the goal is to measure things like metabolomics, proteomics, RNA, all the good stuff that you all love, all the big data that we're promising you, genomics, and the idea is to integrate these different layers for each individual and come up with this amazing diagram that you've seen in Bob Weinberg's talk, to have an understanding of the altered mechanisms at the cellular level. So moving from the tissue and organ level to the cellular level. And yeah.
How can you get from the data, how can you get to the mechanisms?
So the idea is that because we'll presumably have a minimally biased view because we're going to integrate different modalities. That's the promise. We will be able to see and measure many different such modalities on many different individuals, millions of individuals. And we'll be able to measure the physiological range of these modalities and identify deviation from these ranges. So that's nice and good, but we still don't have all of these measurements on millions of patients, specifically not on millions of individuals with ASD. So what can we do with the data that we have today in order to better understand the different disorders that make up the autism spectrum? I want to show you a proof of concept study that we've done, which integrated the whole exome variation with segregation patterns, expression correlation, and sexually dimorphic expression patterns, and electronic medical records and health insurance claims data. So lots of data and prior knowledge, of course, and let me show you what we've done. So we started with whole exome variation, and that is a measurement of the 1% of the functional genome that codes for proteins. And whole exome sequence provides comprehensive survey of functional variation, like, never mind, whoopsie. Anyways, by the time I get this back on, I can tell you that the challenge of whole exome-seq is that we have tens of thousands of variants for each individual, and it's very difficult to infer which variants are related to our trait of interest and which are totally unrelated. So we then use segregation patterns and the idea of Mendelian randomization to try and infer which variants are more related to our trait of interest, which is ASD, of course. So we're looking for variants that are in families, that are shared among affected SIBs in multiplex families. Multiplex families are those with several affected siblings. And then we're looking for variants that are different between discord and SIB pairs. Discord and SIB pairs, one is affected and at least one is unaffected. Additionally, we look at expression correlation. The idea is that we know that ASD and brain development is a complex trait, and so we expect multiple genes and multiple variants acting together and giving rise to some alteration in typical brain development. Next, we focus on sexually dimorphic expression patterns in light of the fact that about 80% of the individuals on the spectrum are male. And finally, we use massive amounts of electronic medical records and healthcare insurance data that we have to powerfully validate our molecular hypotheses. So this is our figure 1. When we had the debate between figure 1 and figure 7. This is figure 1 of our study. And let me show you what we've done. So we start with big data, big transcriptional data, and this is an existing data set called BrainSpan that includes about 300,000 exons from more than 500 samples that are gender, age, and brain region specific, and they create a spatiotemporal profile of the developing human brain, starting from 8 weeks post-conception to adult brain. And you've seen these heat maps in Ami's talk before. That just shows you levels of expression. So we have this massive matrix that we need to analyze, and we use an MIT trick to divide and conquer it. Otherwise, we would have never been able to analyze it. And we identify exons that are tightly correlated in terms of expression. We create a graph from these exon pairs, and that way we identify clusters of co-regulated exons during human brain development, and I will refer to those as exon clusters from now on. Then specifically, we focus on those exon clusters that are sexually dimorphic. Here's an example. We have male expression in red, female in blue, and specifically we care about prenatal brain development because that's when we think ASD, the derailment from the typical developmental program starts in ASD. And we have different ways to visualize it. This is a typical age in log scale. Wait, do you see my typical age in log scale? And here we're talking about functional periods that were defined by neuroscientists. Each period has its own characteristics, and we care most about periods 4 to 7, which are the prenatal periods. So either days 1 to 256 or periods 4 to 7, and we care about big differences between blue and red. All right, and then we take we integrate several whole exome sequence studies because we want to maximize power, and we undergo this very exhausting statistical framework where we try to identify variants within all of these cohorts together. And once we have variants that we can harmoniously compare between the different data sets that I didn't collect myself, I just take other people's data and reuse it for sustainability. We can then identify ASD segregating deleterious variation, meaning, like I said before, different between discordant pairs. This is the affected proband, unaffected proband. We have 1,700 of those, and then those variants that are shared in multiplex families, and we have 50 multiplex families with 2 to 5 affected SIBs in each. Specifically, we focus on deleterious variants, so that would be frame shift, nonsense, and splice site mutations that we think have the greatest effect on protein function. But of course, that's not validated, that's hypothetical. So all of that computational exercise results in clusters of neurodevelopmentally co-regulated, sexually dimorphic, segregating deleterious variation. And that's this hairball is an example of one of these clusters. So how do we make sense out of this? We can ask what is the function of genes in these clusters, and whether any one function is enriched among genes of each cluster. So luckily, most of the functions that are enriched among these clusters were previously implicated in ASD, meaning that are supporting the idea that our analysis is legit, and that includes immune function, chromatin or transcriptional regulation, synaptic function, growth, and morphogenesis. But another function that was not previously recognized was also found in 20% of our clusters, and that's lipid regulation. And let me just show you an example, simple example. So this is a very small cluster of one gene, several exons of this one gene, lipoprotein lipase, LPL, and here we're looking at a plot that shows differences in the deleterious alleles between unaffected siblings and probands. So probands have more deleterious alleles in these exons than their unaffected siblings. And this shows you that exons that are part of this cluster have a sexually dimorphic expression pattern during early brain development. Another quick example, and a simple one, is of another small cluster, LDLR, low density lipoprotein receptor, also sexually dimorphic. And here I'm showing you a heat map that shows several multiplex families in which mutations in these exons that are co-expressed are also shared among affected SIBs of these multiplex families. One last example, I promise. We have this 84 gene cluster that is enriched with a known pathway of metabolism of lipids and lipoproteins. And this is a hairball like the others, also sexually dimorphic during the prenatal periods. And the good news is that now we have a hypothesis to test our data, minimally biased, of course there are some biases, suggests that lipid dysregulation might be a convergent etiology in ASD. So how can we test this hypothesis? We think we know what these genes do. We think that LPL hydrolyzes triglyceride-rich lipoproteins. We think that LDLR internalizes LDL, and that leads to its downstream cellular functions. So what happens if these two are dysfunctional? We predict that that would result in elevated, in higher levels of LDL, in cholesterol, and triglycerides in blood. So that's a hypothesis that we can test. And luckily, I also work at Boston Children's Hospital, and Boston Children's Hospital has decades of electronic medical record data, which we can use to examine relationships between elevated blood lipid profiles and diagnoses of autism spectrum disorder. And that's exactly what we did. We enumerated individuals with ASD and at least one altered blood lipid level that's outside the reference range, and compared that to individuals with no ASD and altered lipid profiles and their complements. So we found that, I'm not showing you p-values, just effect sizes, abnormal LDL levels are associated with ASD odds ratio about 1.4. Total cholesterol is similarly associated, and that means that measurements of elevated total cholesterol are associated with having an autism diagnosis. Similarly, elevated triglycerides are associated. And of interest, here I'm showing you the actual levels just of triglycerides, and we care about some other factors such as gender. We know that there are gender differences in blood profiles. We also care about age. Age also affects lipid profiles. And we care about metabolic status. On the top are individuals with no metabolic-related diagnoses or drugs. And here are individuals with obesity, type 2 diabetes, metabolic syndrome, and related diagnoses or blood test results. And we can see that across these strata, this is for triglycerides, but I can similarly show you for other measurements, individuals with ASD even stratified on these factors that we know affect blood lipid levels still have elevated triglycerides as compared to matched individuals with no ASD diagnosis. Okay, so that's one way of testing our hypothesis, but we always like to use multiple data. We then turn to healthcare claims data. Aetna is one of the largest healthcare insurance providers in the United States, and luckily they shared their data with the medical school, Harvard Medical School, and we can reuse that data to look for relationships between different diagnoses, between spatiotemporal dynamics. We can really make the world out of this data. And so within this data of healthcare claims, we have about 40,000 families with autism, and we can examine dyslipidemia-related diagnoses in these families as compared to families with no record of autism in the family. So we just enumerate the number of individuals with ASD and a dyslipidemia diagnosis, be it hyperlipidemia, mixed hyperlipidemia, lipoprotein disorders, lipid metabolism, all of these good diagnoses, and other disorders of lipid metabolism. So we count them in individuals with ASD, we count them in families without ASD, and we compare them. We can also consider any of these diagnoses, and that provides a very strong association between dyslipidemia and ASD. We have an odds ratio of about 2, which is very strong, and it affects about 6% of children with ASD across the United States. That, 5 minutes? Okay, I'll be quick. So that seems like a very small fraction, but actually it's much larger than any genetic cause of ASD, which affects at most 1% of individuals. To highlight the heritability of this, We can look at enrichment of dyslipidemia diagnosis in mothers of individuals with ASD and in fathers, and they are also enriched, and that supports the heritability of that. Falling asleep? Jet lag? Okay. All right. So to sum up, we can, if we suspect that dyslipidemia might somehow define a subtype of autism, then we hypothesize that it would also be associated with other diagnoses that might be related. And what we did here is we compared all individuals with dyslipidemia and ASD to individuals with ASD and no dyslipidemia. And we find that there are several other comorbidities that characterize individuals with dyslipidemia-associated ASD, and these are predominantly metabolic, so that is consistent with having a robust subtype. But that's all nice and good, but then one of the reviewers said, duh, you know that one of the most commonly prescribed drugs in ASD has some lipid side effects. For example, atypical antipsychotics that are found in about 40% of individuals with ASD, they have a known side effect of causing dyslipidemia. This is from a review showing commonly prescribed drugs and their side effect. And so, of course, we examined relationship drugs. And let me just be quick and tell you that even when we focus on that subpopulation that has dyslipidemia and ASD and absolutely no record of not only atypical antipsychotics but also anticonvulsants that are thought to affect lipid metabolism and anti-diabetics that indirectly affects lipids, we still see an enrichment of dyslipidemia diagnoses in individuals with ASD. We can also stratify blood lipid profiles based on drug use, and still the signal persists. The association holds very strongly. So as Dan said, we then need to ask ourselves, does this theory explain prior observations? And it does. There is a very rare syndrome, Smith-Lemli-Opitz syndrome, or SLOS. This is a very rare Mendelian disorder of impaired cholesterol metabolism that's caused by a loss of function of this DHCR7 gene, cholesterol metabolism gene. It's very rare, as I said, 1 in 50,000 births. And the wide majority of individuals with SLOS are also on the spectrum. So we see a rare example of how impaired cholesterol metabolism might cause or is associated with ASD. We can also go to mice and ask, let's look at mice, at models of mice that try to interrupt a single gene that is implicated in autism as compared to mice that have an interrupted gene that is known to cause dyslipidemia, and examine their phenotypic overlap. To make a long story short, there is a striking overlap, highly powerful. When we try to examine overall similarities in these phenotypes, we see that there is a strong clustering, meaning that overall the phenotypes are similar between dyslipidemia mice in orange as compared to autism mice in blue. Had there been a very strong difference, we would expect that all the orange would be completely separated from the blue. We see that lots of blue, as is shown down here, has nothing to do with the orange, but some blue, some models of autism are very similar to models of known dyslipidemia genes. This DHCR7, the SLOS gene, is shown here in red as an example of a known Mendelian disease that is caused by a cholesterol metabolism defect. How much time do I have? 0 minutes?
1 minute.
What?
1 minute.
1 minute. Okay. Let me tell you about lipids in the brain for less than a minute. Lipids are very important in the brain. They're not only 60% of brain weight, but actually they're essential building blocks of cell membranes. And so you can imagine if I mess up with membranes of neurons, for example, it would mess up neuronal excitability and synaptic function. I can mess up lipid signaling if I have dysregulation of lipid metabolism. Lipids are also important immune mediators, and that would also affect that. Yeah, and now I can relax and speak slowly again. Okay, let me summarize what I've just shown you. We have an integrative work that combines data and knowledge, suggesting that lipid dysregulation may define a specific autism subtype, which is something that we've been trying to do for years. I've shown you what we've done. We started by doing some integrative genomics and transcriptomic study, and that led us to believe that lipid metabolism could be implicated. We then identified altered blood lipid profiles in children with ASD as compared to matched controls. We found enrichment of dyslipidemia diagnosis in individuals with ASD and their parents as compared to matched controls, enrichment of dyslipidemia diagnosis in individuals with ASD as compared to their unaffected siblings, which I didn't show you, and shared neurobehavioral phenotypes among mouse models of ASD and dyslipidemia. And all of that is still strong after stratifying for factors that are known to alter lipid levels. So the implications are, of course, early screening, hopefully, not right now, opportunities for intervention, and most actionably, one step towards precision medicine of ASD, how we might be able to use multiple unbiased or minimally biased data modalities for subtyping ASD. And I want to thank this very good-looking bunch of individuals that did this study. All right, that's it.
So we have 8 minutes for Q&A.
What percentage of ASD do you think is ascribable to alterations in lipid, and could many other ASD cases be operating in which the lipid dysregulation may play a minor contributory role? And the second question is, what if you focused only on X chromosome genes in which the hemizygosity and the X chromosome in males might play a very important role in the sexual dimorphism of the disease?
Very good questions. So this data suggests that about 6% of kids might clinically show meet ASD criteria and dyslipidemia criteria. And you're right that there could be others that have some contribution that we haven't measured and we should focus our attention on. And in terms of focusing on the X chromosome, that's a very good idea. Here we use genetics first to generate a hypothesis and then test it irrespective of genetics, but ultimately our goal is to have genetic data and deep clinical information about everyone, and then it will be one of the first things to do to stratify and focus only on X-linked variation. So, thanks.
Yeah, so I know very little about statistical research, but I wonder, except the autism spectrum disorder is in a behavioral diagnosed. So that means it is a rather vague diagnosis,
yeah,
and it subsumes very different strengths of the disease or of the disorder.
Mm-hmm.
Hmm?
Yeah.
And what you, the cases you're taking to use for the statistics, you treat them all the same, right? Whether
Yeah.
they are strong or not strong.
Yeah.
And how can this vagueness of diagnosis and the exactness of the sequencing or whatever kind of data you have, doesn't it lead to wrong figures?
And by aggregating all of the individuals that are on the spectrum, whether they require more support or less support, whether they're high on the spectrum or low on the spectrum, yeah, that's a very good point. In the previous version of the Diagnostic and Statistical Manual of Mental Disorders, the DSM, there was a distinction between different subtypes or levels of ASD. There was pervasive developmental disorders not otherwise specified, meaning that they are highly functioning, and Asperger's was still in there, and there was autistic disorder, and there was some sub-classification of the spectrum, and it was decided to aggregate them all because we don't really understand where to draw the line, and all the research in the world is based on just taking the big group of individuals that meet ASD criteria and try to somehow subtype them based on other factors. There are several initiatives to try and make sense out of this complexity. For example, there's RDoc, research domain criteria that's initiated by NIMH, where they say, well, let's focus on their brain function. Why do we need to worry about behavioral symptoms? That's one step farther away from the actual problem. And so many different groups and individuals are trying to subtype. We focus on subtyping based on comorbid conditions and genetics. There are many other ways to do so.
So I would like to understand, how do you think about the status of your finding of association between dyslipidemia and ASD? Is the main value understanding the origin of the disease, the prognosis, the treatment, or diagnostic? So
yeah.
I couldn't understand from the way you presented it.
The goal of this is to define subtype that would be targeted by future studies to understand the mechanism, to compare that more comprehensively to other known subtypes such as seizure-associated autism or Fragile X-associated autism, to better understand whether it has a specific clinical trajectory. Maybe we can use blood lipid profiles as an early means of diagnosing and screening these people. So this is just a first step in many ideas for future studies to better understand first the mechanism, and second how we can leverage it to improve the lives of children with ASD.
Unfortunately, we have no more time. Let's thank our speaker.